genetic constitution, and the varies from the basic things, wool and the color. So these Prakriti are basically anatomical, physiological, psychological, sociological variation with the different aspects. Each person, the born with the unique percentage of those character that is determined not just by the genetic bulk, but also by the milieu through the fetal development. Next, ma'am, next. मैम ये आपके अंडर में फिर आगे प्रोसीड करेंगे ना? In contemporary science, at the time of fertilization, the genetic constitution gets established. And on the basis of genetic constitution, there is the preventive, predictive, and the personalized medicine. And the individual having the dependence on the medication, food, situation presented in the way, at the dosic element and the appropriate customization will be delivered to stabilize. There is the step towards the genomic consideration with the different Prakriti, dominancy, Prakriti, and the three those encapsulated foundation of personalized percept for the implementation in the prognostic treatment. It is crucial to create their molecular basis. This Prakriti is basically described in the Dasvit Pariksha as the very popular and the very prognosticate predictive substance, that is the Dosic Prakriti. This Prakriti, what we have learned in the Ayurvedic perspective, that is the Prakritastu Swabhava. किसी व्यक्ति का जो स्वभाव होता है वही प्रकृति कही जाती है, और स्वभाव बनता कैसे है इसके लिए शास्त्रों में वर्णित है। प्रकृति नाम जन्म मरण अंतराल भाव में गर्भाव क्रांति समय स्व कारण उद्रेक जनिता निर्विकारणित उस स्थिति। That is, during the fertilization, the changes in the fetus and the zygote, changing is the genetic constitution, leads to is the particular character. So then the Prakriti determining factor should be either Prakriti, Jati Prasakta, Kul Prasakta, Desanupatni, Kalanupatni, Vainupatni, Pratyatipeta. Basically, these are the factors which influence the genetic character. Originally, they have established during the fertilization of the paternal part and the maternal part. And the Sukrusonit Kalgarbhasa Prakriti. This Prakriti depends on the Sukrusonit Prakriti, that is the maternal and the paternal part. And the Kalgarbhasa Prakriti, nature of uterus, and the maternal Ahar Bihar and the Mahabhut Vikar. These are the aggravating factors. These are factors which influence the characterization of the Prakriti. This dosena adhika aneka nikena vasamanubadhyante means those have the characteristic which is going to be the combined with body, then dosena garvam badhyate. Such type of factor are going to be the influences in the specific zygote or embryo. Now, to understand the Ayurgenomics, we must to know the basic fundamentals regarding the genetics. What is the gene? That is the basic unit of heredity that is passed from the parents to offspring and is made up of the DNA sequences. This is the chromosome, and these are going to be the unrolled, and this red area is called the gene structure. What is the genome? The genome is the entire set of DNA. In human, we are considering 23 pairs of chromosome located in the cell nucleus, as well as the small chromosome in the cell mitochondria. And this genome contains the all information needed for an individual to develop and function. So human being in the 23 pair of chromosome, all the gene have the present in 23 pairs of chromosome, each called as a genome. And what is the genomics? It's the branch of biology that focuses on the evolution, function, structure, editing, or mapping of the genomes. These all component comes under the genomics: evolution, function, structure, editing, or mapping. Now, suppose there, I think so, there is the many MD scholars which have the heard about the gene, allele, SNP, genomes. I am trying to understand, to tell you what are the different patterns, what are the different content. One of two more alternative version of the gene at a specific location of a chromosome, that is called the allele. अगर इसको आप देखेंगे तो यहाँ पर जो हेट्रोजाइगस एलील की बात होती है, दो तरह की होती है एलील, एक हेट्रोजाइगस एलील, एक होमोजाइगस एलील। तो जो हेट्रोजाइगस एलील की बात करेंगे तो दोनों आर्म पर, जो क्रोमोसोम के दो आर्म होते हैं, तो दोनों आर्म पर अगर भिन्न भिन्न जीन है तो उसको हम हेट्रोजाइगस कहते हैं। अगर दोनों आर्म पर एक क्रोमोसोम की सेम टाइप ऑफ जीन है तो हम इसको होमोजाइगस कहते हैं। और अब जैसे इस फिगर में आपको ग्रीन और रेड दिखाया गया है, तो इस तरह से हम इसको लेफ्ट साइड को हम हेट्रोजाइगस और राइट साइड को हम होमोजाइगस के नाम से समझते हैं। एक टर्म आता है SNP, सिंगल न्यूक्लियोटाइड पॉलीमॉर्फिज्म, मींस देयर आर सेवरल वेरिएंट इन द ह्यूमन जीनोम। अगर एक ह्यूमन जीनोम की अगर बात करेंगे तो उसमें बहुत से SNP उपस्थित होंगे। और जैसे इंडिविजुअल 1, 2, 3, 4, उसमें अगर SNP की बात करेंगे तो थायमिन, साइटोसिन, 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 थायमिन, थायमिन, साइटोसिन और थायमिन, ये चार तरह के, चार इंडिविजुअल पर 4 SNP उपस्थित है। दैट इज कॉल्ड द सिंगल न्यूक्लियोटाइड पॉलीमॉर्फिज्म। दैट T and C are going to be the modified in the different as the CC and the TT and the CT. Means one component are going to be the different form. That is called the polymorphism. Morphism means going to be the change with the nucleotide. The second terminology is the transcriptomics. The transcriptomics means RNA transcript produced by the genotype at a given time, but provide a link between the genome. That RNA transcript from DNA to RNA, RNA to protein, and they are going to be the linked by a genome. The proteomes and the cellular phenotypes, they are going to be the linked protein component, cellular structure, and the genomic structure. And the next terminology is the epigenomics. The epigenomics is the branch that leads to epigenetic changes in a cell and the genetic material known as the epigenome. These changes can be caused by age, diet, exercise, drug, chemicals, and other environmental factors. So these are called the epigenomics. Means earlier people are not understanding that is the gene material can be the changed by the environment or the drug or the any other substances. But the branch have the came in the existence that the genetic changes can be made with the diet, exercise, drug, chemicals, and the other environmental factors. The metagenomics means metagenomics is the study of the structure and the function of entire nucleotide sequencing isolated and analyzed from the all organisms. Typically, the specific community of microorganism, जो स्पेसिफिक टाइप का माइक्रोऑर्गेनिज्म किसी कम्युनिटी में अगर उपस्थित है तो उन सबकी जीनोमिक स्ट्रक्चर हम मेटाजीनोमिक्स की कैटेगरी में करते हैं. What is the ayurgenomics? This Ayurgenomics and the integrative approach of Ayurveda and the genomics for discovery of the predictive marker for preventive and the personalized medicine. That is called the Ayurgenomics. And these are the bridges between the traditional medicine and the modern medicine. Means they are going to be the link in new direction. What ancestors have told and the further opportunity to the researcher that what have they told and the what did not have the find till date. That this is the connection between the traditional medicine and the modern medicine providing the vigorous scientific understanding of the basic concept. At the same time, incorporation the practical preventive approaches of the Ayurveda into modern medicine. Now, this genomics is the predictive personalized interpretation of the DNA, tells about the respective individual based on the genetic makeup and the individual in Ayurveda. As the ancient is the personalized medicinal system with the appropriate drug and the dietary regimen. Basically, these are the structure, the genotype DNA, Jamna birth Prakriti, Janma birth Prakriti is the governed by the DNA, mRNA, transfer RNA, and the protein. The total study is the proteomics, and the RNA and DNA the study is the genomics. This mRNA is the categorized is the what in a particular cell, and the transfer RNA is the considered as the pith, and the protein what make a structure and the protection of the cell is the term as the cup. And this epigenetics or protein cup make the change in the genetics, that is the epigenetics, and this phenotype Ayurvedic constitution affected by lifestyle, behavior, as earlier we have discussed in the Jati Prasakta, Kul Prasakta, Desanupatmi, Kalanupat. Or these are the taken from the Sethi in the Parasati Mukherjee in the study I have the taken their reference. And this ancient science of epigenetics assess the status of those personalized diet, lifestyle recommendation to improve a patient's health, and this integrates concept to Ayurveda as a Prakriti with the modern genetic research. It incorporates and the correlates those Vat, Pitta, and Kapha with the expression of specific genes and the physiological function and the fluctuation. The basic concept of Ayurgenomics is to find that those prevents and inherited. Basically, from maternal to paternal, from maternal paternal part to offspring, they are going to be sending some character in the form of the dosa. This Ayurgenomics study the provide a molecular framework of the integration genomics study to accelerate the discovery of marker, predictive, preventive, and the personalized medicine. If we see in the Ayurvedic perspective, the concept of genetics mentioned in the Ayurveda as a Bij part, Bij bhag, and the Bij bhag avayav. Bij as a whole, Bij bhag, it may be the designated as it's a cellular organelle having the genetic constitution, like the mitochondria have the DNA, and the cytoplasm have the RNA, nucleus has the DNA, and the Bij bhag avayav, this chromosomal part it has been taken as the integral in the single component is termed as the Bij bhag avayav. They are deciding the character in an individual person. The Bij bhag avayav. And the Bij bhag avayav, the gene is responsible for the expression of particular trait of an individual. And the Prakriti innate constitution is mentioned as the genetically determined relative proportion of the dosa. The genetic polymorphism within the specific genotype may occur with the different frequency depending on the racial or population factor which evolve from the selective geographic region and the ethnic factor. Desanupatni, there is a study I have been told the genetic differentiation among the Tamil caste is the approximately low, that is 0.96. In having 45 autosomal SAR tender repeat marker reflecting a largely common origin. Nonetheless, caste and the continent-specific pattern are evident. Suppose, for a 32 linkage defining the Y chromosome SNPs, Tamil caste show higher affinity to European than to Eastern Asia. This is the drastically genetically discrimination that Tamil people are more homologous as the African than the Eastern Asia. And the genetic distance estimate to the European are ordered by caste rank, and the 32 lineage defining mitochondria SNP and the hypervariable sequences, Tamil caste have higher affinity to Eastern Asian than to European. These are the studies regarding the country-wise and the region-wise by the Watkins and the Thara the model. This study the earlier described the population identification, the chromosomal and the genomic study, and the Prakriti screening the particular individual, and the Prakriti phenotyping and the biological sample collection, the Ayurgenomic approach done by the genomics, epigenomics, transcriptics, the metagenomics, and the multi-omics sample selection, dispensing phenomena, discovery, and these were going to be performed by the pre-validation and further to the clinical study. It is also published by the journal. Those functions regarding the metabolism. Now, coming on to the point, those journal in human body, like cell division, movement, and the excretion of waste, are mainly governed by the Vata Prakriti. Anabolism, growth, maintenance of structure, storage, and the stability are contributed by the Kapha dose. And the Pitta is primarily responsible for the metabolism, thermal regulation, and the homeostasis. These are the published by the Hangre, a scientific value of the Ayurveda, alternative complementary medicine. It in 2005. Here it was going to be published. Research on the Ayurgenomics, what different genomic constitution and the particular gene we have found. That is the in the very earlier time. A study was established regarding the Prakriti and the CYP2C19 gene polymorphism, that the cytochrome P45C2C9 gene polymorphism is the Prakriti of an individual. And the C19 genotype, the family gene that is involved in the detoxification and the metabolism of certain drug. And what they found in the different variety, like the predominance of extensive of the CYP2, C19 as the genotype 1/1, 1/2, and the 1/3 was observed basically in the Pitta Prakriti, having extensive metabolizer. But the genotypic is going to be the change, and the poor metabolizer of the CYP C19 and the poor metabolizer in the Kapha Prakriti. This evident that the genetic constitution has changed 2/2, 2/3, 3/3. The earlier 1/1, 1/2, 1/3 was the Vata Prakriti, Pitta Prakriti, and the poor metabolizer was the Kapha Prakriti. जैसा कि हमारे शास्त्रों में वर्णित है. And the second time is the PGM1 gene, phosphoglutamate. This enzyme, this gene determining in the fastly found in the different type of Prakriti. And this PGM1 gene is the correlated with the energy production and the more hormones and the constant in the Pitta Prakriti than the Kapha and the Vata. Means the energy production is more in Pitta having SNP-PGM1 gene with the RSID 11208257. This phosphoglucosamine gene is the center of many metabolic pathway like glycolysis and the sucrose metabolism. The function of pitta include digestion, metabolism, and the energy production. This finding suggests that the function of the gene directly correlate with the role of pitta in the metabolism. And the Prakriti and the HLA allele. HLA means human leukocyte antigen. In 2005, the Prakriti are going to be correlated with the HLA DRB1 type. And this HLA complex immune response against the foreign invader such as virus and the bacteria are found more in the Kapha Prakriti. This HLA and the Prakriti typing having the complete absence of the HLA DRB102, HLA dimer allele in the Vata Prakriti and of HLA DRB113 in the Kapha Prakriti. Means HLA DRB113 is the prominent in Prakriti and DRB12 is the Vata Prakriti. In the second allele, HLA DRB110 had the higher allele frequency in the Kapha Prakriti than the Pitta and the Vata. This study has been conducted by the Bettercott, Carvalho, Lee B. Bross, and this very renowned Journal of Immunology Research. And the Prakriti and the cell are complementary system. There are different type of cluster of differentiation of particular cell, that is CD25, CD56, CD14. To find astonished that the CT25 and the CT56 were higher in the Kapha Prakriti and the CD14 monocyte predominantly observed in the Pitta Prakriti. I think so because the monocyte is the most probably the phagocytic and the metabolizer of the organism having in body. This Prakriti and the EGLN1 gene in 2010 studied the high altitude adaptation and the common variation among the EGLN1 gene. There are RS 479200 CNT heterodimer and the RS 48090 TC. These are the heterodimers. They found that the TT genotype was more frequent in the Kapha type and the correlated with the higher expression of EGLN1 gene and the higher incidence of pulmonary edema. Means a person having the EGLN1 gene will suffer the pulmonary edema with having the high Kapha type of Prakriti. In contrast, it was presented a significant lower frequency in the Pitta and the nearly absent in the native of the high altitude. High altitude condition में लोगों में संभवत है EGLN1 की जो वैरायटी है वो absent होती है. The one of their interpretation in the Pitta types are more protected at high altitude. In the Prakriti and the EGLN1 gene in the Wall-Willebrand factor. We see how the change is going on. EGLN1 allele, HAP, high altitude pulmonary edema associated, and the ancestral Wall-Willebrand factor allele thrombosis association was significantly high in the Kapha Prakriti group as compared to Pitta Prakriti. Also showed that genetic link between the EGLN1 and the Von Willebrand factor. Basically, in thrombosis and the thrombus formation, coagulation process, Von Willebrand factor is the very key important role to influence the clotting phenomenon, and which could modify the thrombosis, bleeding susceptibility, and the outcome of hypoxia. The next point is the biochemical and the genomic wide expression level in the different Prakriti. Like Pitta type, there was the overexpression of gene in the immune response pathway. The Vataj mein, there was overexpression of the gene related to cell cycle, particularly in the regulation of cyclin dependent protein kinase activity and the regulation of enzyme activity. So the cell cycle, especially cyclin dependent protein kinase, are abundant in the Vata Prakriti. And the Kapha Prakriti, there is a down regulation of gene involved in the fibrinolysis and an up-regulation of the gene involved in the ATP and the cofactor biosynthesis. Means there is fibrinolysis. Means what fibrin has the made in Kaphas, they have the down regulation of gene and the up regulation of gene involving the ATP. And the Prakriti and the different biochemical marker, that is the Kaphas Prakriti group, had a number of biochemical differences from Pitta or Vata. And the Kaphas Prakriti showed higher digoxin level. Here we are discussing the high digoxin level secreted from the hypothalamic component. That increase the glucoconjugate level, increase the free radical production, and the reduced scavenger, as well as the increase the tryptophan catabolization and the reduced tyrosine catabolization. It means the Kaphas Prakriti have the higher digoxin level, which inhibits the membrane of sodium potassium ATP. So the brain are going to be the decline. They are enhance their sleeping capacity. Thus, this digoxin basically are well for the treatment of the heart failure, but in our body, especially hypothalamic region, is going to be the secreted as the digoxin, and the especially higher concentration in the kapha. The gene expression level and the engagement in the core biological process, such as transport, immune response, blood coagulation in Kapha Prakriti individual. These are the again evidence showed by Parasan again the Agarwal sir, which earlier have the study completed. And the Prakriti and the lipids. This study also suggests that the Kapha type of having the higher level of triglyceride, total cholesterol, high and the low density lipoprotein, and the low high density lipid lipoprotein compared to Pitta and the Vata. Means triglyceride or lipid profile are higher having the Kapha type of Prakriti. The Prakriti and the hemoglobin gene expression. Higher level of expression of hemoglobin gene was observed in the Pitta Prakriti individual as compared to Kapha or Vata Prakriti. You see, Pitta are the homologous and the essential residue in the Rakta Dhatu. And the Rakta and the Pitta are the same in character. Body mass index. The Kapha type of person have the more body mass index and than the Vata and the Pitta type. In the earlier, you see in CCRS pattern, the Kapha type of Prakriti, what BMI will be. These were the assessment criteria. But the study shows that BMI, which person has the higher, having the Kapha type of Prakriti. The Prakriti and the platelet aggregation. In other studies, found that the ADP induced maximal platelet aggregation was maximum among the Vata Pitta type individuals. They are slightly, here we are going to discuss, and the action and the movement was the war. There was the fibrinolysis and the platelet aggregation, but there is the induced and the aggregation and the Vata Pitta type individual. The Prakriti can be correlated with the genetics where there is any particular genetic pattern for the particular Prakriti with the help of a tool like single nucleotide polymorphism P2Y2. The receptor present at the platelet surface initiate platelet aggregation, especially in the Vata and Pitta type of Prakriti. And the Prakriti and the DNA methylation. DNA methylation probably coupled with the chromatin regulation has contributed to the different Prakriti phenotype. And this research provides the insight into the genetics, epigenetic mechanism, and the Ayurvedic personalized system of medicine. The Vatas correlate the lower DNA methylation and the promoter. But the Pittas having the distinct gene promoter, higher metabolism, and the DNA methylation. These are the clarity. The Vatas have the lower DNA methylation, the Pittas have the higher DNA methylation. The study have the made that the whole study was taken 3955 and the 467 MPSRS and the differentially methylated probes related to PMI. What red type is there? That is copper prominent and this higher methylation. And there is the pitta, they are very slightly, very low. And the vata, they are very low. None of there is the are observed as the what type of there is the methylation. These are the distinct variations. Next one is LHX gene. These genes are the comes under LIM domain, and this encodes a member of the large protein family which contains the LIM domain and the unique system rich gene binding domain. This protein, transcribe, is a factor important for the development of the renal and the urogenital system. This gene is the candidate for the candidate having the such type of syndrome, and we can be categorized or can be similar with the univapor disease, where this disease of happening anomaly in the female and the urogenital system. This gene is especially LHX1 gene. Second gene is the SOX11 gene. These are basically prominent in the Pitta Prakriti. What does gene eat in the person? This gene is the intronless gene, encodes a member of the SRY related high mobility group family of transcription factor involving in the regulation of embryonic development and the determination of the cell fate. And also is the developing the nervous system that play a key role in the tumor genesis, tumor formation. The next point is CDH22 gene. The prominent in the Kapha Prakriti. This gene is the member of the Catherine superfamily. This composed of the 5 Catherine repeated domain and the cytoplasmic tail similar to the high conserved cytoplasmic region of the classical Catherine. This gene is especially important role in morphogenesis and the tissue formation. The neural and the non-neural cell during development and the maintenance of the brain and the neuroendocrine organ. And this UTR CDH22 gene, CPG methylation in the island, was also found in the high BMI index, especially in the Kapha Prakriti. The effect of isotonic exercise on the blood response in the different Prakriti type. This diastolic blood pressure immediately after isotonic exercise are going to be decreased within a exercise of 5 minutes, especially in the Vata Kapha style, but does not decrease in the Pitta Kapha in the Vata Pitta style. This Prakriti and the autonomic nervous system activity, the Kapha type of Prakriti have the higher parasympathetic activity and the lower sympathetic activity in the term of cardiovascular reactivity compared to Pitta or Vata. That's why the Kaphas have the tendency to lower sleeping tendency, and the simultaneously in night parasympathetic activity is higher. So the tendency of sleeping is the more in the Kapha Prakriti, the homologous going to the parasympathetic similar to the Kapha Prakriti. The Prakriti and the inflammatory markers. What Kapha type have the higher level of biomarker marker such as interleukin, tissue necrosis factor alpha, high sensitive CRP, and the HOMA-IL. And this IL has the extensively anti-inflammatory function and the role as the myokine. The IL-6 was the first myokine that was found in the secreted in the bloodstream to respond to muscle contract, and the aerobic exercise provoking systemic cytokine response. The tumor necrosis factor is the inflammatory cytokines produced by the macrophages, monocytes during acute inflammation and the responsible for the diverse range of the signaling events within cell. And the high sensitive C-reactive protein is the blood test that find lower level of C-reactive protein. This protein measures the general level of inflammation in your body. This HSCRP can be used to find the risk of heart disease in the stroke. The Vata Kapha and the Kapha Prakriti groups both were correlated with the high level of inflammatory marker. These three most common used inflammatory marker as the C-reactive protein, ESR, and the plasma viscosity. The strong relation of the risk factor, diabetes, hypertension, dyslipidemia, insulin resistance, and the inflammatory marker with the Vata Kapha and the Kapha Prakriti type. Prakriti and the rheumatoid arthritis. Rheumatoid arthritis patient found that inflammatory genes were upregulated in the Vata type, while in the Pitta and the Kapha type there was in the upregulation of the oxidative stress pathway gene. In the incidence of different diseases, Vata Kapha Prakriti has the higher risk for the diabetes mellitus, the earlier we discussed, and the hypertension and hyperlipidemia. Parkinson's disease was the highest in the patient with the Vata Prakriti, and the patient having the irritable bowel syndrome was found the more in the Vata type Prakriti, and the patient with the Pitta having the IBSD or the irritable bowel syndrome with the diarrhea. It also going to be the discriminated the IBS-C with the constipation as a Vata type, and the IBS-D type as a diarrhea type in the Pitta. SOD, sodium oxidation dismutase 3. This is the oxidative stress pathway gene SOD3 and the PON1 observed in the Pitta Prakriti. This gene is the member of the superoxide dismutase family. SOD is the antioxidant enzyme that catalyzes the conversion of superoxide radical into the hydrogen peroxide and the oxygen. Mutation in this gene may be associated with the heart disease. It is the expected in the fat cell, kidney, and the other tissue. PON gene is the member of peroxonose family, and the enzyme exhibits the lactonase and the ester hydrolase activity. This activity is performed in the kidney and the liver and the secreted in the circulation where bind to LTL and the hydrolases. Thiolactones and the gerobiotics, including paraxone, and the metabolites of the insecticide parathene. And the polymorphism in this gene may be associated with the coronary artery disease in the diabetic retinopathy and the heart disease. There are the certain work have been done by scientists, the genomic wise the analysis correlation and the Ayurveda Prakriti. These are the link. There are many genes are present. Genes, position, minor, MF, Prakriti, Prakriti allele, chi-square study, and past in permutation, number of permutations. These are the discussed as a Vatic person, Pathic person, and the Kapha person. The Prakriti and other related factor. In school-going children having the between 10 to 16 years, having the academic stress more in the Vata type of fat mass and the fat percentage was positive correlation with the Kapha type of Prakriti. Those recognize the definition of life and the activity in the body, such as Vata represent input, output homeostasis, with the turnover, negative entropy of the production, and the Kapha have the inheritable structure. These are the specific Vata in the Kapha type of activity. Now, one point is the biorhythm. I am going to the next is critical review. That the biological rhythm or the chronobiology means there are three different types. The what body have the at a particular time going to be the change from 1 hour, 2 hour, 24 hour, 1 year, 6 year, 6 months. All these are going to be the change with the time, and this comes under the three different names as the circadian rhythm within the 24-hour routine changes, ultradian rhythm within for the 24-hour, within 24 hours or the within is the ultradian rhythm, and the infradian rhythm monthly or year means more than that. This various physiological process changing biological clock, biological rhythm, regulating process like sleeping, eating, exposure to light, temperature, etc., cell to tissue, and the whole. These clocks are governed by the master clock, which is situated in the brain, and the suprachiasmatic nucleus. Basically, it is attached with the hypothalamus, and this is going to be the changing with the diurnal and the nocturnal variation, and also amount of light and the intensity of light what they are going to be out. And the circadian rhythm cycle of 24 hours, and also named as the diurnal, and the changes occurring in the daylight, in the dark light, occurring of the 24 hours. And the example of circadian rhythm, what going to be the change? The sleep yoga pattern cycle, body temperature changes, release of hormone, melatonin, cortisol, digestion of the dietary component, and the immune function. We see in the circadian rhythm within the 24 hours at the 2 a.m. there is deepest sleep, and the 4:30 the body temperature are going to the very low, and the 6:45 sharp rise in the blood pressure. 7:30 secretion of melatonin ceases, and the intestinal movement begin at the 8:30, and the maximum secretion of testosterone. And now these are coming in the daytime, in the midway, and this is the coordination at the best at 30, and the coming into the pran reaction time at its the best. The muscle strength that affecting the cardiovascular disease, maximal blood pressure, body temperature, reduction of melatonin begins. Basically, these are governed by the genetic constitution what the body has, finding there the amount of light and the amount of darkness. This ultradian rhythm, course of 24 hours, occurring more than once a day. This cycle is repeated throughout a 24-hour circadian rhythm, a circadian day. This contrast with the infradian rhythm, there is the recurrent period occurring longer than a day. In ultradian rhythm, the cycle was a period shorter than a day and longer than an hour. Activity, the arousal, high performance, sleep, ultradian, healing response, high performance, stress. This is 90 minutes the high performance, 20 minutes ultradian healing response. These are the consequently occurring as a cyclic manner. Some ultradian rhythm, this is the rest activity cycle, stage of sleep, blood circulation, pulse, heart rate, hormonal secretion, thermoregulation, maturation, bowel activity, nostril dilation, appetite, and the arousal. These are the ultradian rhythm. The infradian rhythm lasting for the more than 24 hours, but shorter than the 1 year, occurring weekly, monthly, and the annually. Several infradian rhythms are known to be caused by the hormonal stimulation or the exogenous factor. Suppose, example, the seasonal depression and the example of infradian rhythm occurring once a year can be caused by a systematic lowering of the light level during the winter season. And the second one, the consequences at the menstrual cycle is occurring. It is an example of infradian rhythm where this particular type and the day of cycle and the possibility to conceive, ovulation of the fertile window, possible to conceive at this duration of time, and the thickening of uterine lining and cycle of the menstruation. These are in the example of infradian rhythm. These are the genetically determined. The person having this sequentially easily or not easily going to be the done. The some person or the some female have the problem with the menstruation. The some are going to be the no. So these are the certain example of infradian rhythm. This Ayurvedic perspective, the biological rhythm as the Visargadhan, Vinchepo, Somsule Nilayatha, Dharanti Jagade, Kapit Nilayatha. Consequently, in the cyclic pattern, we people are knowing what in the annually, 6 monthly, yeah, every 2 months, the season or the dosas are going to be the changing. Accordingly, the body are going to be the changing. Suppose we are called as in December and the January, the person temperature are very low. The why body are going to be the save for that. The body in the have the anti-freezing substances which protect from the severe coldness, and some animal having the residing in the very deep, yeah, very minute, very cold region, and in the minus temperature region, they have the anti-freezing substance. We are going to be produced by the genetically determined substance. They are going to be protected even the very severe cold. And the circadian rhythm and the ultradian rhythm in the Ayurveda and the ahoratri and dieting component. We are taking it as the in 24 hour, in the 12 hour, and what changes going on. Diurnal variation, if people are knowing, and switching over then the next one. This molecular basis of circadian rhythm. What happened basically easily earlier was in 1980, a gene is the fruit fly the isolated that encodes a protein that built away the each night only to be the broken down in the following day. In subsequently work, the trios, 3 scientists, as the other scientists, picked up the molecular regulation of the period that is called the PER and identify additional component of the circadian clock. In 2017, Nobel Prize of Physiology Medicine awarded by the Geoffrey Hall Rosbus in the Michael. The discovery about the molecular mechanism that controls the circadian rhythm. The 3 scientists again going to be the elaborated the establishment of the transcriptional translational feedback loop, the production of the protein in the night time and the going to be degraded by the daytime at a particular gene, that is the CYS and the BMAL gene, that are going to be the degraded when they are going to be the completed their signaling pathway in the time in the cell. These are the work performed as a Drosophila and the mammal. Earlier was the Drosophila and the cytoplasm, this nucleus component. There are PER gene, TIM. We are going to be the signalate, the clock and the cycle. The PER gene going to perform the protein structure, and these reside in the cell. And this PER and TIM gene are going to be the loaded this protein inside nucleus. So at a particular time, when the duration has completed of the in night time, this TIM protein are comes in the cytoplasm, and they are going to be degraded by the DBT gene, double gene. This double gene are going to be degraded. Same way in the mammal, there was the cycle gene, but in mammal there are BMAL1 gene, and thus PR gene are changed as the PR13 and the CRY12. In the Drosophila, they found the TIM gene, but in the mammal there was the CRY gene. The same phenomena, the PR gene and the CRY gene are going to be uploaded this protein in the nucleus, and the particular when the awakening, they are going to be released in the cytoplasm, and the CMA same, the AEIE, the kinase enzyme are going to be degraded this PR CRY gene. Now, coming the conclusion point, what the different Prakriti have the different specific gene are abundant. Now, still there is so many work as going on, but I'm trying to make it some Prakriti and the particular specific gene. The Pitta Prakriti, earlier we had the CYP2C19 PM, and the Kapha Prakriti have the CYP2C19 PM, that is the poor metabolization and the extensive metabolizer. The Pitta Prakriti have PGM1 gene, and the Vata Prakriti have HLA DRD0102, and the Kapha Prakriti have 013 and the 010 gene. And the Kapha Prakriti is defense mechanism CD56 and the CD25. And the Pitta Prakriti has the natural killer cell CD14. And the Kapha Prakriti also has the EGLN1 TT genotype. Also in the HAPE with the 1 milligram factor, high altitude pulmonary edema. The Vata Prakriti are dominant in the cyclin dependent in the protein kinase, and the Kapha Prakriti, the down regulation of the fibrinolysin and the higher hypothalamic digoxin level. This lipid profile, high BMI, and the CD22 genes, and the higher parasympathetic activity are more in the Kapha Prakriti, while lower DNA methylation and LH1 gene in the urogenital deformity more in the Vata Prakriti, while SOX11 gene and more in the Pitta Prakriti, and the high level of HB gene expression were more in the Pitta Prakriti. So this the overall discussion, overall study, the mobility and the activity-based gene is prominent in the Vata Prakriti. The metabolic activity and the gene metabolic activity gene base prominent in the Pitta Prakriti, and the defense system and the immune-based gene is prominent in the Kapha Prakriti. So there is a step towards the genetic field, and the overall still there is challenge to genetic deformity of this analysis. We are going to be the studies the majority of the extensive single type of Prakriti that are going to be the prominent a particular gene, but we have to do this Dvanda Prakriti according to is the character of the gunas present in the individual person. I think overall this concept, I as a Carl Sagan, a scientist, so many comments have there, but I got, you have to know the past to understand the present. So many criticisms over there, but more than work has been done regarding the different scientists in the field of Ayurveda. Still there is going on, and there is the golden era of Ayurveda till now. And thank you for the presence here. Hello.
Yes, sir. We are here.
जी मैम.
Just a minute, sir. एक second.